It's a pleasure to be here. I am going to present this for the first time, so you're the recipient of my trial and error, I guess. So this is going to be about joint cancer ecology models, a name that I just came up with based on some other inspiration. But anyway, these are probabilistic methods for population-level tumor microenvironment analysis. Again, so first of all, and this is going to be a disclaimer mostly. I am at Biostatistics, and I do Bayesian statistical models as my job with full uncertainty multiplication, and my background is in statistics and economics. Read no biology. And so therefore, when I'm going to present some of the results, you should probably ask my collaborators. Ooh, there you go. My collaborators for more information about the interpretation, although maybe I can say something. I don't know. We'll see. And today we'll present, in this presentation, there's mostly going to be about a story about joint cancer ecology models, but the tool that I'm presenting is BIPS. Where is it? Here. BIPS is our package that you can all use for multiplexed imaging data analysis. And the work that I'm presenting is also mainly done by Joel, who used to be a PhD student in bioinformatics, and now he's a postdoc at Hopkins. Let's begin. So what is the tumor microenvironment? And this is something that I've come to learn in the last couple of years. We have a spatially structured set of like cells and tissue elements, and the cells are of different types, right? So we have like immune cells as well as cancer cells and other kinds of cells. And the key idea here is that the way in which these cells organize spatially, so they occupy this environment, depends on how other cells occupy the environment, because maybe they interact with each other, because maybe there's like some regulation of things, at least in my understanding. And so the way that they arrange is dependent on the way of all other cells arrange. And again, the life cycles of these cells are influenced by each other and therefore also by their spatial location. And I'm also mentioning that treatment may impact all of these dynamics. And so whenever we say anything about the spatial structure, we may ask the question, does treatment or say cancer stage impact this spatial structure? And we are going to focus on multiplexed imaging data for the tumor microenvironment, or TME. And so the question is, we have all of these features. How do we create a model, a statistical model, that translates all of these features into math, basically, right? And then once we have that, we all know that with statistics, we can do hypothesis testing and essentially do science. So let's dig in a little bit deeper, right? So the first thing is that TME is spatially structured. And so here on the right, I have an example from an actual TME image in which you see that there's the expression of some protein, right? And we see some spatial structure. What do I mean? In statistics, when we think about spatial structure, it's essentially the idea that everything is related to everything else, but near things are more related than distant things. And related, I just mean, if you look at this image, for example, this is a simulated spatial data set. Things are pixels, right? And pixels that are close to each other are more similar than pixels that are far from each other. So that's essentially what we mean. And we contrast this with an IID assumption, which you may be familiar with, which is essentially the idea that there's no relation between any of the pixels. And so there would be like no spatial structure, there would be no pattern in space. And in this other image, not what I wanted to do. In this other image, the relationship is we see similar colors again, similar in the location that they are in space. And if you go farther away, they're less related to each other. And by the way, if you have any questions during this presentation, feel free to interrupt me at any time. The other thing that we want to mention is that the TME is populated by cells of different types. And, we have like maybe some phenotyping pipeline, but essentially they are all occupying the same space. And therefore, they are also related in the way that they are occupying this space. Of course, our goal is to analyze not just one cell type at a time. We want to analyze to give a complete picture of the entire set of cells in this tumor microenvironment. Because again, because of their relationships. And studying just one at a time would like kind of miss the relationship that a cell type has with others. And so here's another example in which we have, in the same data as I was showing before. These are proteins now, so there's like before phenotyping. I think I used the raw data in this case. But the idea is that you have many proteins that you see that may be related to the cell type, right? And all of them have a spatial dependence, because you see that the points that are close to each other tend to be more similar than points that are far from each other. And maybe the way that these proteins are related to each other, we can see that there's a spatial dependence, right? Maybe because a few protein types have similar values in similar locations. So this is like sort of the idea. The environment, as in any environment, and I'm referring here to the cancer environment, but it could be any natural environment too, we have environmental features, right? And so these are describing the conditions in which these cells live. And in any model that we want to develop, we may want to explain how things vary depending on these features, right? So in the natural world, we may have climate change, and so that what happens after we have climate change, what would have happened in this environment if we didn't observe climate change? That's the sort of question that we ask. And these, in the TME, could be treatment, cancer stage, other sort of variables. And I'm like referring to them as subject level. So they are constant across the entire image because they relate to the subject to which that image corresponds to. Or FOV. So the FOV is like the image of the tissue that we're taking in this specific example. And we may take multiple images of the same tissue, but they are constant across the image. So there may be a gradient across the tissue. And then pixel level covariates, or cell level in other terms, where we have information specifically on each of the cells. For example, the categorization, so the cell type is an information that we're going to use. And others may be like, the size, or I don't know. You will have to tell me because I'm not that expert. But anyway, we may have information about the cells. And then the other key feature, and this is something that distinguishes cancer data from natural sources, like natural environments, is the fact that we can collect multiple TME images, right? We have multiple patients. And the patients, again, because of the information that we can collect from them, these create cohorts of groups of individuals that we take images of, and we may want to figure out what's the signal in these images that makes all these patients similar to each other, and what's the noise, what's just patient variability. And the other important aspect of these images is they cannot be aligned, right? So if I take an image from this patient and an image from a different patient, I cannot overlap them, and they're not referring to the same sort of tissue because they're simply different patients, right? But still, we may think that because all of these images are collected from patients with PDAC, then some of the ways in which these cells organize with each other are similar. And maybe some of them are just related to noise or patient variability. So a statistical model should quantify all of these things, which, many, right? The first one is how much spatial dependence there is for every cell type, right? So this is the degree, in a sense, of spatial clusterings of cells of type A, say, and then we do the same for all the cell types. So we may find that, in some images, the orange points tend to be close to each other, and in some, they tend to be not close to each other, right? And so, but we want to quantify how likely it is that for two cells of the same type to be together. And then we also want to control and measure and quantify multivariate dependence. So we say that we have controlled for all of the spatial dependence, all of the spatial pattern, then what are the associations, the residual associations between cell types? And this may tell me something about interactions, right? Although we may have a discussion of what it means for cells to interact relative to occupied space, and I'm open to questions on this. And the other thing that you want to do is not just how every cell type clusters in space, not just how there is association between cell types after controlling for spatial dependence, but also the cross-cell type spatial dependence. And so that is to say, if I observe a cell of type A here, how likely is it that I'm going to see a cell type B next to me, right? And so that is the degree of spatial co-clustering. You can imagine this is also important when maybe we want to see whether treatment changes the way in which immune cells go and attack a cancer cell. I mean, that's my understanding, right? And so that's co-clustering, cross-variable spatial dependence. And so all of these we want to do together, right? We don't just want to have a model for this, a model for this. We want to control for how much is the spatial dependence of one cell type after, like we basically attribute the different components of the data, the variability of the data. We want to say this much is due to clustering of the same cell type, this much is due to multivariate dependence, this much is due to cross-variable dependence. And then all of this, in all of these components, we may want to have covariate effects, right? So we want to use the covariates that I was mentioning earlier, so these ones, explain how things are changing in space. For example, treatment could be like this 0-1 dummy covariate in which we're explaining how the organizations of cells of type A, they begin to not cluster as much, maybe. Or the association between immune and cancer cells improves, something like that. We want to explain all of these things with covariates. And again, all of this is because we want to distinguish what is common structure, so the signal, from noise. And again, I will never fail to mention this. We want to do a joint model, which means that we want to do all of these things at the same time. We don't want to study just one aspect without also controlling for all others. And when I say that we want to statistically quantify things, I mean that we want to be able to run tests, right? We want to run hypothesis testing, controlling for uncertainty, and essentially this means that we want to do science, and we want to really be able to say, this is not just random noise, this is signal that we have observed in this data set. Questions so far? Good. And so BIPS is the software package that does all of this. All of the things that I've mentioned, you can do with BIPS. So it will quantify the spatial dependence of every cell type. It will quantify the multivariate dependence after controlling for spatial dependence, and it will quantify cross-variable spatial dependence, and it will do covariate effects in some form. I mean, not all of the possible ways. That is kind of the future plans. And then essentially the idea is that we'll find some signal relative to noise. And you can find this on GitHub. It's in our package. So let's go to the application, right? So here's what we want to really, here's how it actually works. So we have multiple cohorts of patients, right? So we have, for example, the patients that have been diagnosed with a certain disease, say IPMN, introductory neoplasm, that. Then there's another patient cohort that has PDAC, some it's a worse disease, right? Like adenocarcinoma or something for pancreas, right? And then we may have other cohorts, and we collect several images, right? So every patient may have one or more images of tissues from their biopsy. And what we do is, you will observe that these are point patterns, right? So the point pattern is a spatial data set in which we know the locations of objects. And this is a multivariate point pattern because we have the locations of objects, so cells, of different types, right? So we color code them. And so you see that the orange cells cluster in this area, and then these like sort of like tissue cells cluster in different ways. And the idea here is that we will want to say, what is different between IPMN and PDAC, right? What's the spatial characterization of both that we can understand how they are similar and how they are different. The first thing that we do, we have to do in these settings for the purpose of fitting models that we're implementing via BIPS is we transform each image into a count gridded raster image, right? So for example, I think here I'm taking, let me go with, okay, I am taking image 347-2, and you see that there's like multiple cell types. And so we take a grid, right? We count how many cells of that type there are in each small pixel, and then we create a count multivariate data set that is like this, right? And so for example, the ABC cells, darker colors correspond to larger counts. And here we see that that should correspond to here. We see that there's a cloud here that corresponds to having this here, right? And so on and so forth. And we do this for all of the other cell types. And this is a pre-processing step that we need to run before we run the analysis. BIPS does that. And so then what we do, and I'm having this as a very non-math sort of presentation, so I have some slides, but not much. What we do is, and which is what BIPS does, is it fits a Bayesian log Gaussian Cox process to multi-subject, multivariate points pattern data, right? This is like a little bit of a long sort of thing, right? So the idea here is that, I don't know if any of you is familiar with a Poisson process. Is anybody familiar with the Poisson process? So a Poisson process in one dimension is sort of like telling you when events happen, in time, and there's like an inter-arrival time and all of that. This is like a two-dimensional Poisson process where you don't have a time, you have a space, and you know the locations of where events happen in space, and then you have multiple event types, and these events are just the cell types. You see where cells occur. These models are very difficult to fit. So the first thing you usually do is do that sort of operation of transforming the data into a gridded count data set, right? So we have count data with which correspond to this idea of modeling a two-dimensional Poisson process. The idea here is that we observe the counts, and we think of the counts as arising from this Poisson distribution, where the intensity, the mean of the Poisson distribution is the smooth spatially varying surface. And so here I'm giving you an example of a smooth spatial surface that could be the model for the intensity of this Poisson process. What does it mean? It means that if the value is high, like in this area, the intensity of the Poisson process is high, and therefore I'm expecting more cells of this type in this location of the spatial domain, which is the tissue. Conversely, if the image gives me low data, then it is unlikely that I would see this cell type in these locations. So every point, every cell type will have a point pattern. Every point pattern corresponds to a latent, so unobserved, of course, intensity surface. And we want to relate the intensity surfaces with each other, explaining how they're similar and how they're dissimilar, and therefore give you all the things that I was mentioning earlier. Here's another explainer. So here, for example, cells of type 1 on the top left, right? So we see the counts, and here this is simulated data. And here we see the intensity that we had used to generate this data set, right? So larger values correspond to more likely events in that region, and vice versa. Lower values, like for example here, correspond to less likely events here, right? It's the same connection. The idea is, based on data that looks like these two, like we're given these two cell types, we want to recover the intensity, not because we care much about the intensity, but because by recovering the intensity, we are also going to be able to say how similar they are to each other. Like if you look at these two patterns, it may be difficult to say how these two are related to each other. But if you look at these two, sorry, no, not what I mean. But if you look at these two intensity maps, you may look at, there's maybe a positive correlation between these two cell types, because here we have large values, also large values here, large values here and here. And there's sort of like an area where there's less probability. So we don't care about the specifics of the intensity, but the fact that these two may be similar to each other. And how similar are they? What do we mean when we are quantifying spatial similarity? And this is the point of correlation model. Yes, please.
This one. So how did you convert from these demand on the left to the intensity?
So this is simulated data, meaning that first we generate this, right? We create a map, and then based on the value of this, we sample a Poisson random variable at every pixel. And therefore, if the mean of the Poisson is large, it is likely that this count pixel is going to be larger. If the mean of the Poisson is small, then it's likely that the count will be 0. This is just a simulated data set. The point is, we are going to get this, and we are going to use BIPS to estimate this, right? Like we are going to recover that. And in the paper, I took this from the paper that I'm going to cite at the end. In the paper, there's a third column, which is the recovered intensity map from BIPs from the simulated data set, right? We give BIPs this data, and what image do we recover for the intensity? You will see, I didn't put it here, but it's like sort of like very similar in the way that the patterns are recovered. So when I mention spatial dependence, what do we mean, right? So spatial dependence, we characterize with the study of correlation functions. A correlation function is basically telling me how similar, right, these 2 points, for example, are with each other based on the distance that there exists between them. In spatial statistics, every point in this is the realization of a random variable, and therefore, when I say how similar two random variables are, I compute the correlation, right? And the correlation as a function of distance, you would say here, it's pretty strong because these points are very similar to each other. They have a similar color, so the similar realization, but I can imagine that the random variables are related to each other. And then the more that I expand my distance, the correlation goes down. But here it goes down sort of like it decays slowly, right? So there's this parameter that's called spatial decay, tells me how slowly the decay occurs. And this is slow, and therefore we get a lot of spatial dependence. Strong correlation, even when we are quite far from in the spatial domain. Conversely, if we have no spatial correlation, then we would recover a correlation function that drops very quickly, right? Because there's no correlation now between variables that are this far. I can go very close and still there's not going to be any correlation. This is sort of like white noise. White noise would just drop from 1 to 0, right? As soon as you increase the distance to any positive number, that's 0, white noise. And so the idea here is that we're going to be able to produce not just a correlation function for every cell type related to the intensity that we're here, right? So the idea is cell type, estimate the intensity, measure how much this is correlated, sort of like what we're doing here. And then we're going to be able to have a curve for every cell type. But then also we are going to say, well, based on the similarities of these two random variables, and they're like at this distance, right? You see the points can be similar. I can draw a curve for any pair of cell types too, right? So we have lots of correlation functions to estimate. And so BIPS does all of this, right? So BIPS will give you one correlation function for every pair of cell types across the entire cohort of patients. So the idea of BIPS is we can't just use, in spatial statistics, you would be able to fit this data, right? And get a function for the correlation for one image, one correlation function. But when you have cancer data, that's not really useful because then we have many correlation functions for many subjects. And what is signal? Like what is common between these correlation functions and what is noise, right? And so instead, what we do with BIPS is we estimate one correlation function for every pair of cells for the entire subject cohort, right? So if you are in the PDAC cohort, you're gonna get a correlation function, and then if you are in the IPMN cohort, you're gonna get a different correlation functions. The idea is that whatever I cannot explain with this correlation function is subject-specific heterogeneity that we did not account for with a common structure. And then again, we can divide these correlation functions into two main types. The first one is when we compare cells of the same type, and those are called marginal correlation functions. And then when we compare cells of different types, those are called cross correlation functions. And in the future, something that BIPS does not do currently is we want to introduce some covariates in the way that we explain how these functions change, right? So you would have a covariate maybe saying that if you are, I don't know what continuous level, there's a continuous treatment variable maybe, and then that makes the correlation function change smoothly. That would be the ideal. Some technical notes, BIPS runs entirely on R. So you don't have to do any sort of like advanced processing or whatever. It offers pre-processing the way that I was explaining it earlier, and there are online vignettes. But then all of the operations are done in C++, right? So we want BIPS to be like efficient and scalable to large data settings. Under the hood, BIPS implements some of the best models for scalable spatial inference for multivariate data, because these data are not actually simple, right? We have count data, so non-Gaussian, which means that if we want to be Bayesian and do posterior inference with this data, we're going to have to use some complicated Hamiltonian Monte Carlo type methods. And the other thing is we have multivariate data, so that adds to the complication. We have spatial dependence, which is typically very difficult to account for with methods, but BIPS is pretty good at doing all of this. And it will give you full uncertainty quantification in the form of samples from the posterior distribution you can use to do quantiles. I'm not sure how many of you are familiar with MCMC output, but you can compute all of the intervals, all of the predictions that you want based on samples from the posterior. That you don't have to do any, for example, sort of bootstrapping, cross-validation, all of that. You don't do that because you're Bayesian, you have your priors, you update your priors with the data, and you get the posterior, and then using posterior, you can do whatever you want. And it's the joint posterior, so it will give you all of the dependence in all of your unknowns. It's the same methods that BIPS implements in other contexts compare favorably usually with competitors. So they usually have better compute times, better efficiency, better predictions, all of that. And so here's, I'm gonna work through the analysis that we did on the PDAC versus IPMN subjects, right? So here's an example of 2 images from IPMN and 2 images from PDAC. And you see that maybe because PDAC, I'm not sure, this is just my understanding, because PDAC is a more cancerous sort of disease, there's a lot less structured dependence as opposed to IPMN. Right, so these are the inputs. We give this to the pre-processing functions in BIPS. And what we get, for example, this is from that one image, the 909-1, we get, as I mentioned earlier, the count for every cell type, right? So here, darker colors means that there's higher counts. Then using that, as I was saying earlier, we have a count map, and then at every location, so the counts for subject I, for cell type J, at location S, so right now, this is one image, so I, for the subject, cell type 1, so J is equal to 1, and then this is location S. This is a modeled as a Poisson random variable where the intensity must be positive for a Poisson random variable, so we put the exponential, right? The intensity is then, the log intensity technically, is eta ij, so this is something that varies in space, where every subject gets its own, right? So subject I gets the intensity, subject I and cell type 1 gets this intensity map, where at every pixel, we model this as changing depending on covariates, and then the spatial factor model that gives us the spatial dependence. What is a spatial factor model? This is a linear combination of univariate Gaussian processes. So once you take this is a vector technically. So this is a vector of k univariate Gaussian processes. Collectively, they are a multivariate Gaussian process where the parameters, and this is the key is that the parameters theta, as you see, they're not indexed by I or J. Parameters are common for all subjects in the same cohort. There is, though, once we have this Gaussian process, the image that we see that is not common across subjects. So it's basically saying the spatial dependence, every subject gets its own, but the way in which we produce it is common across all subjects, right? And then the linear combination here is just related to the fact that we are taking this cell type. And again, these are subject level covariates. We may have like something more complicated here. I just wrote a simple way to simplify notation. Otherwise, we would have had like xi of s and so on and so forth. Any questions on this structure? Okay. And then the application specifically, we have 143 PDAC patients, 89 IPMN, and we have 6 types of cells. Then we process each raw image of the point patterns into 70 by 70 pixels, right? So raster images. We would process them the way that, not this way, but the way that we process them before the analysis is like doing 70 by 70 in the application. The total runtime for the entire estimation and model fitting is about 7 hours. You may think that that's a lot, but that actually computes to about 10 minutes for full uncertainty quantification for every pair of correlation functions, which is not much, right? We have 42 of them, right? Because we have the 6 marginal and the remainder cross times 2 diseases, right? So there's like quite a few things that we need to compute. And this, again, including uncertainty quantification. And so this is the output for when it comes to the actual correlation functions with uncertainty, right? So the bands correspond to our measurement of uncertainty, how uncertain we are about where they should sit. And so here we are looking at the marginals, right? So for example, for Treg cells, we see that PDAC has a lot more clustering of Treg cells, which means that they tend to be close to each other relative to IPMN. Whereas for this tends to be the message here, right? So for these immune cells, there tends to be more clustering of them in the PDAC cohort relative to the IPMN cohort. And this is marginal, right? So we are looking at just one cell type at a time. So this is how one cell type clusters with other cells of the same type. And except for this is a mistake. These are all cross covariance correlations, right? So this tells me how, for example, let me see, APC and T helper co-cluster in space in PDAC versus IPMN. And here's a somewhat interesting find, I think, is that although the way in which APC and T helper co-cluster is very constant in space, meaning that they tend to still have like strong correlations later, at zero distance, so the way in which they associate immediately close to each other, it is actually stronger with IPMN. And this is not possible, for example, to infer if you, I mean, although there's like not much difference between that, statistically speaking, if you didn't do uncertainty identification, you wouldn't know where to put the band, you wouldn't know whether these two are actually different from each other or not. The other thing that we can extract, based on all of these correlation functions, we can say we take the value at zero, right? Just the value at zero, and we make a sort of like this correlation plot for the two diseases. And we see, for example, that epithelial cells tend to be more negatively correlated with others at zero distance in PDAC relative to IPMN. And then we can also do something else, which is we can do the compute the differences, right? So based on these plots, and all of this doesn't require any additional computation. We just take samples from the posterior, right? We take these plots and we compute the differences with the bands, right? And we do that and we find which pairs of cell types. So, we look at the off-diagonal, off-diagonal elements, even the diagonal. This is telling me how different the two diseases are, like the spatial correlations between pairs of cell types are between the two diseases. And for example, there seemed to be not much difference between the PD-L1 CD8 cells and them, and the same types of PD-L1 CD8 cells tend to not be very different in how they occupy space in IPMN relative to PDAC. And I believe that none of this you really can do easily if you do single cells analysis, single cell type analysis or single subject analysis, or if you don't do a spatial sort of model-based approach, and as opposed to doing something like more simple, like a descriptive statistics sort of approach. So that's it, actually. I'm kind of early. So some future work. First of all, I need to credit Joel. So Joel is responsible for completing this like whole article that you can find here with like more information about this simulation setup that we had and everything. He was like really great to work with. I wish. And then in the future, again, I want to implement more complicated models where we can have covariates explain all of these differences. Right now, essentially what we're doing is we have a single covariate, which is whether a subject belongs to IPMN or PDAC. And based on that covariate, we have like basically the whole like differences between correlation types. But we may have like more complicated covariates. There's also more data where we have like, we collect multiple images from the same tissue of a subject, and we know where those images are in the tissue, and then we want to maybe find like a broader multi-resolution gradients based on that, but that's all future work, and for now, this is all I have. So that's it. If you have questions, feel free, and also my email, so anytime. Yes.
you have an iteration for the like, number of samples with balance like this?
Number of samples, you mean number of subjects?
Yeah.
You can technically run this with one subject. Like the entire field of community ecology in statistics runs essentially these models, some variant of them. They're called joint species distribution models, and they run on one habitat. And typically, they may be the best, like the more complete models, they may run the entire Earth data, right? But we have one subject that is Earth, and here we can do much more. Like what you get in those models typically, when you do one subject, is that these curves become wider. Like the uncertainty about these curves becomes wider. Whereas here, because we have 100 subjects, 143 for PDAC, and about 90 for PMN, we tend to have lots of information to estimate these. And of course, when we submitted the paper to journals, the reviewers was like, well, but why can't you, because of this, you can technically estimate a global correlation, cross-correlation function, meaning all of these pairs, and also a subject-specific cross-correlation function. But then that becomes like way too complicated because of the fact that you don't have enough data. So in terms of how many, I feel like in simulations we've run even with like just 20, and it works really well. I mean, you can run it with however, but technically even just one. Because every pixel is technically, you can use data from other pixels to estimate the correlation function. Then of course, it comes down, I feel like the question is more like, how representative is your sample of the entire population of PDAC patients, right? And if you have a sample of one, maybe you can still do something, but do you trust it? Depends on the. Yes.
Yeah, thank you for that. I'm just curious about TLAC, as you can see in the previous slide.
The what?
TLAC regulates with cell.
Okay.
It's very binary cell type. So the cell type abundance is very sparse. So then you cannot easily trust 0 is really 0. So the prediction from the TLACs or other minor cells can be somewhat unpredictable or.
Yeah, so, and this is, I will pick up this slide now. So let me give you an interpretation. Like I don't really know, but from a statistical perspective, you're saying there may be cell types that are very sparsely observed. For some subjects, we may not even see them, right? And so that is where this sort of formulation actually helps. Because if you have, say, 15 cell types that you want to study, and you're using less than 15 spatial factors, even if you were doing more, but essentially the idea is that you can borrow information from other cell types. Maybe you have seen another subject in which that cell type you observed and it was related in some way, and so the subject that doesn't have a lot of information is not going to inform the computation of the correlation function as much. That's the key idea of having multiple subjects, is precisely the fact that some subjects don't have much data. I mean, with some subject with very, very sparse data, we have to remove from the data set because there's literally nothing we can do. But when subjects have, say, very few cells of one type, that's still informative, right? Because that still tells me, here I'm showing correlations technically, but what we compute in the model is covariance, right? And so if you have very few cells of some type, the variance of that cell type would be very small. Then we still want to interpret the correlation in the sense that it's more interpretable. But that's a fair question, and it's precisely where this sort of model helps, because you borrow information across subjects. So even if, again, a subject doesn't have many cells of one type, some other subject may have, and you borrow information across cell types. And so if you've seen that these two cell types are correlated with each other in some other subjects, you're going to infer that they should also be related in this. And this sort of where also the Bayesian sort of methodology helps, because the idea is that, even though subjects are all different, they are arising from the same sort of mechanism. We want to believe that, for example, all of the subjects with PDAC have a similar sort of like stage. Otherwise, they would have not been diagnosed in the same group. Does that answer your question?
Yeah, thank you.
Yes.
Really cool analysis. For sure. I'm curious if you can talk a little bit about the sensitivity to the choices of scale and sort of size.
Yes.
There's a number of places where it might enter in, like how you did the rasterization,
Mhm.
and even number of spatial components, or like fire over the Gaussian process?
Yeah, those are all very relevant questions. We have all of that in the paper. So the discussion typically is how fine should you make this grid, how small the pixels? So because these pixels are essentially approximating a point pattern, the log Gaussian Cox process. And so there's very many actually directions that you could go. Like if you take these too big, so if you take this too big, you're losing the you're essentially unable to measure the correlation function that is beyond the dimension of that pixel. But if you go too small, then you are essentially failing to really estimate the intensity because you have like one count for every cell, and then you never see. It will be more difficult to estimate the correlation at zero spatial distance because at zero spatial distance, you never see anything together, right? And so it is all like sort of like a sensitivity analysis. And in the paper, we try different options, especially with simulated data. And we find that typically you don't really need like ultra high resolution grids to really approximate the true quote unquote functions. The other thing that you can choose here is, so this is a spatial factor model, meaning that if you have, say, 12 cell types, you can choose from 1 to 12 spatial factors, and that's a choice. In the software, we are asking the user to choose that. And so we did some sensitivity analysis, and typically, if you choose too many factors, then performance of MCMC, is degraded because there's many more things to estimate. If you choose too few, then it is going to be more difficult to distinguish between different curves for different patient cohorts because there's less flexibility. And so typically, again, a midpoint is typically what works best. What else? The other thing is, oh, yes, in future work, and so I guess we don't actually have to ask the user to specify the number of factors. There are many methods in statistics, in Bayesian statistics, where you can dynamically learn the number of factors based on data. But again, it's sort of like the thing that we don't think gives you really like it takes way more time to code something like that than it takes the user to just fit a different value and try for themselves. Does that answer all of the question? Yes.
So this method is tested on the image-based spatial transcriptomic data, and so it's ultimately in your correlation class, you are looking for the distance, right? I was wondering, so if there's another spatial transcriptomic acquisition, for example, like a sequence space, like a slice or slide tape, in which each cell has its spatial coordinates. Can you convert it can we like maybe like one of the directions for your future work? Can you like take this kind of input data and then also convert it to something similar to this?
So if I answer the question, so the data that we've used was proteomics data, right? We have proteins, and then you phenotype into cell types, and then you count where cells are. This specific package will work with count data. So if you use transcriptomics data, if you have, I would say, with transcriptomics data, you probably have like hundreds or thousands of genes, right, at every location. It may be a little difficult to fit that data to this. Like we may need to, because this is sort of like doing more than you would be able to do with that sort of dimension. You can still fit, but you would have to wait a longer time. So no guarantees that it's going to only take 7 hours. The alternative thing, I have another paper in which I'm not taking the counts, but I'm taking the expression levels of proteins. So in that case, maybe you can still fit a Gaussian model instead of having this like sort of model for count data or point pattern. If you can fit a Gaussian model conditional on the locations of cells, that's much easier to fit. Because Gaussian makes everything easier, but the complexity here is basically only like having multiple subjects. For transcriptomics, the dimension is usually the problem, and so if you wanted to do something similar for say, instead of having the location of genes of thousands of different genes, you would definitely have to like run this factor model, let's say here, where you're choosing still like a very small number of factors. So even if you have thousands of genes, you would be able to only use like say 20 factors, which maybe it works, right? In many situations, you know, spatial factor model is basically like a different way to say PCA with spatial processes, right? So if you're looking at your data in a preliminary step and you do PCA and you see that the first 25 components explain most of the variability in your data, then you can probably use these models because you would fit them with about 10 to 15 factors. Does that answer your question at all, someone? I hope so. Thanks. Yes.
So this is a parametric model, so you could also picture a comparable non-parametric model where you would take, say, pairs of cells and ask how far apart they are.
Yes.
And then you could create like a pair correlation function just directly from that empirical distribution. Can you comment on why you went this route as opposed to that route and maybe some of the trade-offs?
Yes. This is a semi-parametric model in the sense that the process we're using is a Gaussian process there. The idea here is that these models are much more flexible for future developments if you wanted to actually include, for example, covariates, right? And so as opposed to computing the, say, G-cross function for two cell types, like you want to explain how that changes or how that is common. And so it is somewhat related, right? Because you can still use those methods and you can run, say, the G-cross for every pair. This gives you like a complete picture of all pairs of cell types together. And you can still do many of, I mean, if not all of the things that you can do. Where is this? Like that would be, let me think. So if you're modeling every pair of cells and you're looking at the neighbors, I feel like that's a sort of method like that would kind of fail in the question that I've had earlier when some subjects don't really have many cells of some types, and you would see that those curves wouldn't really be curves, right? It would be like very like stepwise, blocky, and you wouldn't really be able to explain that. I guess if you pull them together for all subjects, you would have like a cloud of curves. Although that doesn't really tell you, like, if you're computing the cross between two cell types, it doesn't control for presence of other cells, right? You're just computing it marginally for two cell types, but you're not considering the fact that there's maybe a third cell type also, whereas these models do that all together. But yeah, no, it's fair. I mean, it's another thing. Joel, by the way, so the former student, we also have another paper which kind of goes more that direction with a model-based approach for modeling pairs of, for interactions essentially. And then you're looking at, how those curves change, but based on a model, as opposed to just computing the empirical G-cross functions. Does that help? Thanks.
Question.
Yeah.
You assumed like this Cox process or Cox response model that has a strong assumption that the variance and the intensity are identical. But oftentimes you can have like a negative binomial or something where you would have over dispersion. And I was curious if you tested that or at least assessed whether or not that fitting over dispersed model would be more explanatory.
So this is a very interesting question because I have another student that we're working on completely different data set, right? So we have marine ecology, marine biology data. We have like sardines, right? No, different species in the Mediterranean Sea. And she was interested in modeling that as a zero inflated Poisson model, right? And she's like, come to me last week saying, hey, Michele, but I've tested the Poisson model versus the ZIP model, and they perform quite similar. And the reason is that, yes, it is completely true. And in this case, probably more valid. Your question is actually more valid. But if you have, the idea here is, okay, so suppose that this is very negative, you're going to see a lot of zeros here, for example, right? And so how will you reconcile the fact that you see so many zeros with the fact that you're assuming a Poisson where the mean is equal to the variance? But the point is that, so let me give you another, maybe a better way. How would you address sort of like overdispersion in a Poisson model for IAD data. The way that you would do that, you could do that, is by writing the log intensity as covariates plus white noise. Right? You would add white noise, and if you assume that that white noise is gamma, that is a Poisson gamma Bayesian model, right? This is actually doing the same thing essentially. So we don't necessarily have a lot of reasons to implement something that is not simple as a Poisson, but it is completely possible. In fact, I think BIPS actually does have the option to implement a negative binomial. I don't know that we tested it. The reason why your question is actually more relevant in this setting is because we have multiple subjects. And so maybe we have evidence for one way or another, which would be otherwise very difficult to have with a single subject data. But it's a very relevant question. And again, the point is, we can have lots of zeros here in a Poisson model where at other locations we have many counts by just adjusting the spatial surface that we have. So conversely, if we observe many zeros and some clustering of the counts, then we can attribute that to a low intensity at those locations. Thanks for the question.
Maybe just one last question. For the MTMC, you did an MTMC.
Yes.
Did you also consider doing like a variational Bayes type of model fitting?
Not in this setting, just because the way that BIPS is implemented is it's an extension of a package that I had before, and that was in MCMC. I am testing currently a different model for different data for different everything in variational inference, and I mean, yeah, it works really fast. However, when it comes to these, so these are like zero distance spatial correlations. That model in that setting, which is a little different, no complaints there, but it does tend to quite significantly underestimate uncertainty in those settings. And so, it's sort of like good to figure out like the broad picture, and then you maybe want to still run MCMC to get the full picture.